so we're talking about cardiovascular pressure control systems, which sounds very fancy, but it is a way to talk about how blood pressure is kind of controlled, regulated, changed in the body, and what causes that. So we're talking about here regulation of arterial pressure, which when we talk about arterial pressure, we're really just talking about blood pressure in general, because arterial pressure is where we measure it. Now, we know that we also kind of downstream of arterial pressure, we have like capillary pressure, which we usually refer to as like hydrostatic pressure, and you don't have the fluctuation up and down that you have in the arteries. So in the arteries, when you measure that, you have a systolic and a diastolic. Meanwhile, downstream, you don't have that. But really, we're talking about blood pressure. So the aorta divides into the major arteries serving each organ system, and you can kind of see those here listed. So brain, heart, wall, kidney, liver, bone, skin, muscle, other. So these work in parallel. So if you reduce, so we can see that with heavy exercises, this bright green here. So when we start to exercise, we can see that vasodilation occurs in the muscle, which causes that more pressure or more blood flow to the muscle and less to like the liver or the kidney. Okay, so that's kind of what's going on, is that arterial pressure to each of those is controlled by those muscular arteries that can constrict or dilate to kind of reduce or increase resistance. Okay, so arterial pressure is cardiac output times total peripheral resistance. We have this term here, TPR, total peripheral resistance, and that is resistance to flow. Okay, so remember when we talked about let's see, the Poussey equation, and we talked about how if you have a vessel diameter and you decrease that vessel diameter, you increase it by the radius to the fourth. Okay, so really by doubling something, you increase the flow by like 16 times, you reduce the resistance by 16 times. So changing that diameter has a huge effect on this total peripheral resistance. Now, total peripheral is a total, and so that's going to be the resistance to flow. And really, what we're talking about is pressure. Okay, so if you increase that total peripheral resistance, you increase blood pressure. Okay, and that kind of brings all of those other equations that we looked at together. Kind of pulls a lot of that in there. Okay, so arterial pressure has to be tightly regulated, okay, and we're going to discuss two primary systems, and then we're going to discuss some others like vasopressin or antidiuretic hormone, and we'll go through several other types, local control, that kind of thing, chemoreceptors. But first, we'll start with these two bigger ones, or they're bigger ones. We'll just leave it at that. So the baroreceptor reflex is so that's a reflex, okay? So that's why it's called a reflex. It's a reflex that will measure blood pressure in the periphery and then will send a signal to the brainstem to either activate the sympathetic or the parasympathetic division to regulate that blood pressure. So we're starting here with detection, so we're kind of figuring out, so this is where we're gonna do the measurement. And so it's the whole system, rapid acting, neuronal mediated reflex, functions to keep that arterial pressure constant through feedback to that sympathetic and parasympathetic nervous system. So kind of what I just said. And of course those work primarily by vasoconstriction, vasodilation, but also on the heart with rate and contractility. Okay, so how hard the heart pushes, contractility. All right, so let's kind of look at how this works. So the baroreceptor, blood pressure is constantly detected in the walls of the carotid sinus. So I have here a picture of the carotid sinus. It is right here in this area. So this where the common carotid artery splits into the external and the internal carotid. Okay, and that's where, and you can kind of see in this picture, it kind of shows that nervous tissue here. And then the how it just kind of forms up here with the glossopharyngeal nerve. Okay? So we see this nervous tissue, there's going to be mechanical receptors. They're going to kind of join up, yeah, like with this, with the glossopharyngeal nerve, and then that's going to go off to the brainstem. Now the aortic arch, I don't have a picture of it here, but if you can kind of picture your heart here, and then coming out of the artery or the aorta is the aortic arch. So we have detection there in the aortic arch. And what they do is they measure stretch. Okay, so we have to measure blood pressure. The way to do that is with seeing how stretched it is. If those mechanical receptors say that it's stretched very much, well then that indicates that the pressure is higher. If it's stretched less, that's going to say lower pressure. Okay, so stretch receptors detect stretching of the arterial wall, and like I just said, increased stretch is more activation, so increased frequency. Because remember, that's how neurons say that something is more or less, is by increasing the frequency, and then less stretch is a lower frequency. So that's kind of always kind of going. That change, or that input is always coming into the brainstem, and then a big change is noticed, and that's going to then feedback. And so we'll kind of talk about how that happens. So baroreceptors are highly sensitive to those changes, or a rate of change in that pressure. Okay. So I have a note here that the changes in receptor sensitivity, or set point, is often present in people with chronic hypertension. So, you know, here are this whole baroreceptor reflex that's supposed to be controlling blood pressure, and then you have people with blood pressures, you know, in the 180s systolic. So obviously it is not, its set point is wrong, or the sensitivity is wrong. Something is telling the body that it's okay. Now, we've also talked about this resistance to flow that can happen with arteriosclerosis, kind of hardening of the arteries. They become less compliant, and so you have to push harder to get things through. But we also can see that there can be a sensitivity change in the baroreceptor that it's just not conducting right. Okay, so speaking of conduction, so this is conduction. So how does the signal get from the carotid artery, or from the carotid sinus, or the aorta, to the brainstem? And we already saw on the carotid sinus that the glossopharyngeal nerve is what's carrying that information, carries it from the carotid sinus to the brainstem. So that's cranial nerve 9. And then the vagus nerve carries it from the aortic arch to the brainstem. So both of them move to the brainstem. One is through that glossopharyngeal, and then the vagus nerve is the one from the aortic arch. So the cardiovascular centers are located in the medulla primarily, but also kind of moves up into the lower part of the pons. Remember, it's medulla and pons; they're right there together. There's a location, or a region in the medulla called the nucleus tractus solitarius. That's where the information is integrated, and then the information, high-low, inhibitory, activating, or excitatory signals, then will either send signals out to the sympathetic or the parasympathetic. So we have a flow chart of that here. So carotid sinus, cranial nerve 9, glossopharyngeal, to this nucleus tractus solitarius. So if we see that it is an excitatory signal, so we have rapid innervation, or rapid signaling, frequency, that means there is stretch. Okay, and so that's probably something to keep in mind. So increased activation equals stretch, okay, which equals high pressure. So when we see this rapid movement here, then or this rapid frequency of neurotransmitter of action potentials, then it's going to activate the parasympathetic system, which is going to slow the heart, and can also decrease contractility in the atria. Okay. On the other hand, if we have slower frequency action potentials, okay, so this decreases, that's going to indicate lower pressure, so everything is flipped. Okay. So it's going to be lower pressure, which is going to then activate, again, goes through that NTS, the nucleus tractus solitarius, So it moves through that, and then it goes to a couple of brain regions, the cardiac accelerator and the vasoconstrictor. So now we have vasoconstriction involved, and that's going to, both of these things will increase blood pressure. So now we have increased frequency at the SA node, increased contractility of the heart, and you have vasoconstriction of the arterioles and the veins. Okay? So another kind of look at this, the way this is taking place. So if high blood pressure is detected, the signal goes to the medulla, so we can kind of see it here through that glossopharyngeal, if it's the carotid sinus and the vagus nerve from the aorta. And then the release of acetylcholine, which is parasympathetic, at the SA and AV nodes. Okay, so we can see the vagus nerve is going back. Of course, these would be together, but it's going back to the AV and the SA nodes. And then, that's spelled wrong. And the heart rate's going to decrease. All right. So then low blood pressure response, remember that's lower frequency. So that's going to be norepinephrine to the SA node. So that's going to move through the sympathetic chain, and then that's going to go to the SA node. But then you can also have activation of the adrenal medulla to release epinephrine, and that's going to go through the bloodstream then, and that's going to affect contractility of the heart. And it's going to, this is saying that it's also going to activate vasoconstriction and of the veins and the arteries. Okay, so I was saying that it goes through the blood vessels, but yeah, it does. It's just not shown in that picture. But then you have vasoconstriction, okay, and that's kind of different here from what happens up here with the parasympathetic. And parasympathetic is slowing the heart, but this and reducing contractility in the atria primarily. But then for the sympathetic, you're also getting stroke volume. You're changing contractility of the ventricles, and you're increasing stroke volume, and you're increasing, like I said, those vessels, those blood vessels are constricting. Okay, so example of baroreceptor function in response to hemorrhage. Hemorrhage is a rapid blood loss. It could be from trauma, it could be from anything, but you have internal bleeding, anything like that. But you have rapid blood loss, and so you're going to have hypovolemia, so you have less blood, and you have less hemoglobin overall too, because you have less blood in general. But your reduction in blood volume is from that blood loss. So you're going to detect that. If you have less volume, you're going to have less stretch. Okay, so that's going to affect those baroreceptors. They're going to notice that difference in stretch, which means you have lower arterial pressure, okay, because the aorta and the carotid artery are arterial. So decreased stretch, which is going to be that decreased firing rate, so slower action potential rate. And then you have the decreased parasympathetic activity of the heart. So we're saying, okay, parasympathetic activity is going to be shut down or reduced, which is going to allow the heart rate to increase. And that's, of course, by increased sympathetic activity to the heart and the blood vessels. Increased heart rate, increased contractility, and increased constriction of the arterioles. And, of course, we denote that with total peripheral resistance. And then constriction of veins. Okay, so remember, veins, we call that unstressed volume because it's under low pressure. So most of your blood is kind of contained in the veins at any one time, and that's kind of a blood reservoir. So if we're looking up here and we're saying, okay, this patient is hemorrhaging blood, they're losing blood, one kind of convenient thing to do would be to squeeze those veins and then get that blood, instead of just sitting in the veins, really not doing anything, slow volume or slow flow, then we're pushing it into the functional system. So yeah, it was functional, it was moving slowly, but now we pushed it into the arteries, and we're going to increase pressure and increase perfusion. That's what we want, is we want to perfuse the organs. Okay. All right, so long-term regulation of blood pressure is going to involve the fluid volume and kidneys. So one thing we need to know, that first bullet here, that fluid volume is directly related to blood pressure. If you have more fluid in your body, you're going to have more pressure. Okay, so most of that, now you can say that, well, it could be interstitial. Well, it could be, but it's also going to be in the blood. So you're going to have higher pressure throughout. So, I mean, you could think that just drinking more water could increase your blood pressure, and that's true, but you know, your kidneys are going to try to push that out. So this is a way that the kidneys are actually involved in increasing your fluid volume. So high fluid volume increases pressure. That's why we give fluids when blood pressure is low. It's primarily, like I said, controlled by the kidneys. So too much blood volume will increase. So if your blood volume is too high, okay, so that could be measured by stretch, that's going to increase urination. So the kidneys are responsible for making urine. So if they produce more urine or allow more blood so that plasma gets filtered through the kidneys, and then that can leave as urine. So your kidneys have that ability. So if you don't have enough blood flow, or if your pressure is too low, that's going to reduce urination. The kidneys are going to say, okay, nope, we're not going to allow as much to be filtered through the kidneys, because you know, we don't want to lose urine. So the big way that we're going to talk about this is the system called the Renin-Angiotensin-Aldosterone mechanism. We're also going to talk about, I mentioned before, Vasopressin. There's something called atrial natriuretic mechanism, or atrial, that involves the atrial natriuretic hormone, and we'll discuss that. But first, let's talk about this RAAS, or the RAA system. So the renin-angiotensin, and that's angiotensin 2, aldosterone mechanism. So let's start where it starts. So you constantly have blood moving through the renal artery into the arterioles of the kidney, and then that will move into this area called the glomerulus, where there's a this capillary ball. Okay, it's in this thing called the Bowman's capsule. I don't know if you remember, but there's this little ball of capillaries that kind of filters blood, and it puts that filtrate right into the kidney. Okay, it's kind of, you know, kind of shaped like this. So we have arterioles going in and out, and then blood is moving through this, and then let's see. So we'll say blood is moving through so this is the glomerulus. I'm just going to draw it again. Okay. So it's a ball of capillaries. You have arterioles going in and then out. And from here, you have blood cells that will remain, but you have, and I'm going to make it yellow. I don't know how easy this is going to be to see, but then you have the filtrate that kind of moves into these nephrons. Okay. This is a very poor picture, obviously. But then the filtrate, this is fluid from your blood that moves into the nephrons. So this is yellow because that's kind of what color your plasma is, and the red blood cells don't go into the kidneys. You shouldn't have red blood cells in your urine. So that's what we mean by filtration, is we're filtering out larger proteins and red blood cells, and we're just putting in here, we're putting in that filtrate, which is going to become urine. So it's going to go through and around, and then it's going to come down to collecting ducts. I ran out of room here, but then you're going to have urine. Okay, so a lot of stuff happens. We'll cover it all when we talk about the kidneys, but the point is that you're constantly kind of measuring the pressure of that where it's going in, and so when the pressure is low, this you will reduce an enzyme. It's actually an enzyme called renin. Okay. So you reduce renin like renal, and that is going to start the show. That's what it's called, the Renin-Angiotensin-Aldosterone mechanism. So the renin is produced or is released by low blood pressure. And then renin breaks down something called angiotensinogen. Okay? Angiotensinogen is made in the liver. It's in your blood right now. It's just going around, just waiting, to be broken down into angiotensin 1. Okay, so ogen, or the gen at the end, means, I don't know, I always think like generates. It's going to generate something else. So this usually means it's a precursor to something else. In this case, angiotensinogen is turned into angiotensin 1, which is then turned into angiotensin 2. Okay, so let's kind of go through that. So renin turns angiotensinogen into angiotensin 1, and then we have this molecule here, angiotensin converting enzyme, that converts angiotensin 1 into angiotensin 2. Angiotensin 2 is active. Okay? This is the active form. All right, so started with renin, angiotensinogen to angiotensin 1, and then you have ACE, which is angiotensin converting enzyme, converts it to angiotensin 2, and then we've got a whole slide talking about all the things that angiotensin 2 does. I have a note here that it's a potent vasoconstrictor, so this is increasing blood pressure, okay? Which makes sense because we started out with low blood pressure. So we activated this system, and now we have vasoconstriction, and a whole lot of other things are actually going to start happening. So angiotensin 2, so it's a potent vasoconstrictor, but it also stimulates the adrenal cortex to secrete aldosterone. Remember, the adrenal cortex sits up here like a hat, or the adrenal gland sits up here like a hat on top of the kidneys. The cortex is the outer part, the cortex is the outer part. The medulla, remember, that's where cortisol and epinephrine are released, is the inner part. So now we're releasing this molecule. So the angiotensin 2 causes this adrenal cortex to release a molecule into the blood called aldosterone. Okay, so aldosterone is what we would call a mineralocorticoid. So it's a hormone that's made by the cortex. So aldosterone, it acts on the kidneys by telling it to reabsorb sodium. Okay? So if our little nephron up here, we're saying, hey, we're going to put in these little sodium transporters, and we're going to get more sodium out of the cell. Okay? And of course, chloride's going to follow the sodium. And what else is going to follow the sodium? Water. Okay? So remember, so now water is also leaving. All right, so what's happening is that we're reabsorbing the chloride, we're reabsorbing the sodium and the chloride and the water. It's all going back into the body. So whereas this was going to all become urine, okay, it's all going to go through the nephron and ultimately become urine down here. Now it's fluid back into the body. So we've reclaimed fluid. And now we're not going to, now that fluid's going to be in the body, which is going to increase the pressure. So as a result, urine volume decreases, so we're going to have less urine flow, and blood volume increases, and that's going to cause blood pressure to rise. Okay? All right. So, let's see. Okay, so there is something else. That's why I'm kind of pausing, because something else, and while I have this terrible, awful picture here that maybe you can figure out what it means and maybe you can't, hopefully you can, but something else happens when angiotensin is released, and that is that it will reduce the filtration rate. So it kind of squeezes on these arterioles and says, not so much blood is going to be moving through here now. So now the blood isn't even getting an attempt to become urine. So that filtration rate, the rate at which things are filtered through this glomerulus, and there are of course millions of these, is reduced, and so we have a reduction of even the possibility of making urine at that point. So not only are we reabsorbing what's there, but we're also kind of limiting how much is even filtered in the first place, and that's what I think this next slide is going to say. Let's see. All right. Well, we'll come across it. I don't know. So angiotensin 2. So these are the effects that angiotensin 2 has. Leads to the release of aldosterone from the adrenal cortex. Stimulates thirst. So thirst is important because that means you're going to increase volume in a more direct way. You're dumping it into your body. Increases pressure. One of the most potent vasoconstrictors known in humans, so that's in humans, so it's going to increase blood pressure, increases sympathetic output to the heart and blood vessels, which is going to increase cardiac output, and it also stimulates the release of something called antidiuretic hormone, which we are going to talk about. Okay. All right. Let's see. So I'm just looking. I think I might be thinking of something else when I'm talking about the increase in filtration rates. So we'll come back and look at that. But this is a flow chart of the RAA system. I hope that didn't just get confusing. So this is the flow chart of the RAA system. Decrease in arterial pressure, release of renin-angiotensin to angiotensin 1, and then the angiotensin converting enzyme converts angiotensin 1 into angiotensin 2. So I have a note on the other page kind of pointing out that ACE inhibitors, those will reduce how much angiotensin is produced. So this is kind of always going on. It's tonically active, and it just kind of gets turned up and down. So if you inhibit the angiotensin converting enzyme, you're reducing the amount of angiotensin 2. And if you reduce angiotensin 2, then you reduce all of these effects here. Okay, so, and what were those effects? So increased aldosterone, so sodium reabsorption, water reabsorption is flowing through, and then more sodium reabsorption, increased extracellular volume, so you've kind of turned up the sodium hydrogen exchanger. That's not something we really talked about before. All right. And then the increase in thirst and vasoconstriction. Okay. So increased total peripheral nutrition, or total peripheral resistance, and then I have this note over here. I don't want to really go into the whole thing. You can just kind of read it. But ACE2, angiotensin converting enzyme 2, actually breaks down angiotensin 2, so it's going to have kind of an opposite effect. It produces something that has even more of an opposite effect. Promotes vasodilation, which is going to reduce pressure. Also, it's anti-inflammatory, and this ACE2 is a purported entry point of COVID-19. So that kind of made the news a little while ago, and increased the amount of research to it for it. All right. So antidiuretic hormone ADH, vasopressin is also called vasopressin, released from the posterior pituitary. It's a diuretic that increases urination, and urination decreases fluid volume in the body. That makes sense. Okay. So if a diuretic increases urination, that's going to decrease fluid volume because you're urinating it out. Well, antidiuretic is going to do the opposite. So this antidiuretic hormone, ADH, vasopressin, same thing, is released from the posterior pituitary, and it is going to cause you to reabsorb more water, maintain more water. So what's that going to do to pressure? It's going to increase it. Okay, and it's kind of in the name, vasopressin. So both of these names kind of give us a lot of information. Antidiuretic, okay, it's gonna cause us to urinate less. Vasopressin, it's going to cause vaso meaning vessel, pressure. It's going to increase pressure in the vessels, which means it causes vasoconstriction. And that checks out, has two main receptor types, so it has its effect in two ways. V1, vasopressor receptor 1, causes vasoconstriction in the arterial and increases TPR, total peripheral resistance. And the V2 increases reabsorption of water into collecting ducts of the kidney. Now that's what I have a picture of over here. So remember, this is our nephron. So this is where we kind of had urine flowing through there, and now some of it, so we were talking about reabsorption of sodium by aldosterone, so that was going on kind of in this distal tubule. Well, then that moves into something called the collecting duct. So that moves into the collecting duct. And then in the collecting duct, either you're going to reabsorb water and put it back into the bloodstream, or you're not. Okay? And that is controlled by vasopressin, ADH. Okay? So ADH will cause more aquaporins, okay, to be inserted, which is going to allow more water to move through. So remember, most cells have these aquaporins, and most cells are permeable to water. This is a little more controlled. So in this collecting duct, if we don't want water to be reabsorbed, we just don't put in aquaporins. And that water has to move through, and that's going to be high urine volume. Well, with vasopressin says, we're going to put in a lot of aquaporins, and we're going to reabsorb a lot of water. Okay? So our urine is going to be very concentrated, so not much urine. Okay, so, and of course that moves its way into the bloodstream, which is going to increase pressure. All right, so promotes reabsorption of water. Water is thereby not lost in urine. So if the question is, if ADH increases reabsorption of water, what's that going to do to blood pressure? It's going to increase fluid volume, therefore it's going to increase blood pressure. All right, so this is the one that increases glomerular filtration rates, the atrial natriuretic peptide. So hopefully we're going to remember that, even though I just didn't. So we just finished up with antidiuretic hormone. Now we're going to talk about this ANP, atrial natriuretic peptide. There are other types of natriuretic peptides. I have a slide kind of explaining what they do in general, but they're really, natrium is referring to sodium. Okay. So they're going to, one of their primary things is to cause this dumping of sodium. So we're going to dump sodium out of the body, which is going to lower pressure because we're causing an increase in urine excretion. Okay, and so if you think about it, this is the first one that when activated lowers blood pressure. Now the baroreceptors, they can go either way, but this one is kind of known for reducing pressure, so remember that. So, like I said, there's atrial natriuretic peptide ANP. There's also something called B-type atrial natriuretic peptide, or BNP, which is actually more of a lab value that's measured in the lab to kind of indicate things like heart failure, indicator of heart failure. And let's see, I think there's a CMP too. I don't know why I didn't put that up here. Probably would be more confusing. So let's talk about how this works. Low pressure baroreceptors are also located in veins, atria, and pulmonary arteries, so keep that in mind. In this case, we're talking about pressure baroreceptors in the atria. So the atria of the heart. So remember, the atria are the top parts of the heart, and when they are stretched more, then they send a signal to the kidneys, and it tells the kidneys to start, not to reabsorb sodium, not to reabsorb water, and to dump those, okay? So release from cardiac muscle cells when atrial blood pressure increases directly enhances sodium and water excretion, and it also inhibits ADH, because if ADH is there's always some level of activity of ADH, but we're saying, okay, ADH, forget it. You're out of a job for just a little while. And remember that collecting duct, that water that was being reabsorbed through those aquaporins, if we block ADH, that water stays in the collecting ducts and is released as urine. Okay? Those are urine droplets down there. Okay, so it inhibits the release of ADH, so it kind of goes against that. Remember, ADH was trying to increase pressure. We're not letting it. decreases blood volume, obviously, because it's all leaving, or much of it is leaving as urine. Okay, high urine output. Renal vasodilation. That's what I was trying to draw. I was hoping I didn't have to draw it again because I'm such a terrible drawer. But that glomerulus, remember, so we have in and out. So where we're filtering blood, and that glomerulus, we can constrict that and say, okay, less blood is going to be filtered. Okay, and then that goes on to the nephron. This is going to make more sense with the after we study the renal system, but we're constricting how much, the easy way to put this is we're restricting how much blood is filtered into the urine or into the kidneys. So we're reducing urine flow or blood flow to the kidneys in general. So that means less is filtered, that's glomerular filtration rate. So we're decreasing that glomerular filtration rate. When glomerular filtration rate is high, we're increasing glomerular filtration rate. When, I'm doing all kinds of dumb stuff. So when glomerular filtration rate is high, you have more going in. So I'm just telling you everything just opposite. It's opposite day. Okay. So if we're increasing glomerular filtration rate, then that means more urine is flowing in. Okay? Or more blood is flowing into the glomerulus, which means more filtrate is moving into the kidneys. Okay? Which means that ultimately you will have more urine being produced. Okay? All right. Whew. Okay. So it increases your GFR, your glomerular filtration rate, and that higher GFR leads to increased loss of fluid through urine. Because remember, that's why I keep getting mixed up, because this one we're decreasing pressure. Okay? So pressure is lower. We're losing fluid. Okay, it also counters the counter-regulatory system for the Renin-Angiotensin-Aldosterone system. Again, the RAA system normally increases pressure. If we block that, then we're again decreasing pressure. We're not allowing the RAA system to increase that pressure. And then I just have a note down here that BNP, this is the B-type atrial natriuretic peptide, has there have been papers done that show that it is an accurate indicator of heart failure, so increases of that. So let's talk in general about what natriuresis is. So a natriuretic peptide is natriuresis is the loss of sodium and water. Like diuresis is increase in urine production, natriuresis, the loss of sodium. And sodium and water is going to follow that sodium. So natriuretic peptides promote salt and water excretion. So let's kind of go through this flow chart again, just to make sure I'm saying everything right. So increased blood volume causes increased atrial stretch. So that's on the atria of the heart. Okay, up here. Myocardial cells stretch and release natriuretic peptides. And then the natriuretic peptides have their effects in lots of places. So, again, they go to, remember the posterior pituitary is so the hypothalamus, it's going to tell the hypothalamus to release less vasopressin from the posterior pituitary. It's going to tell the kidneys to increase glomerular filtration rate, so increase how much blood is filtered through the kidneys. That can even become urine. Okay, urine. Okay, it's going to decrease the release of renin, which slows the whole renin-angiotensin-aldosterone system, and it's also going to tell the medulla directly to decrease blood pressure, which is going to increase the parasympathetic mechanism. All right, so the heart, I mean, the heart has a lot at stake. It knows if there's like too much blood that it's gotta kind of reduce that. So the ultimate the ultimate outcome of this is increase in salt and water excretion. So all of these things are going to cause an increase in salt and water excretion, and then and then decreased blood pressure overall. Okay. All right, so we're kind of repeating ourselves here, if that's okay. Natriuretic peptides antagonize renin-angiotensin-aldosterone system. So this is kind of going through how that works. Enhanced sodium, yeah. So this is kind of saying the same thing that we've just gone over. Okay, so other ways to regulate blood pressure. So chemical responses. So one other thing we know, so the baroreceptors were saying, oh, if it's stretched more, it's going to increase, that means increased blood pressure. Well, one other thing we know is that if oxygen levels are low and carbon dioxide levels are high, that we probably need to move blood more quickly. We need to cycle blood through more to become oxygenated, and we need to kind of increase pressure, increase that movement of blood through the body. So we have chemical receptors. So peripheral sensory receptors are sensitive to oxygen. So this is peripheral. That's what we want to keep straight, because we've got peripheral. And then down here we have central, which central nervous system. So peripheral is outside the brain and spinal cord, so it's all the stuff out in the body, and central is in the brain and the spinal cord. All right, so they're in the carotid and aortic body, so kind of in the same place that we saw for the baroreceptors. So but they're sensitive to chemicals, so sensitive to low partial pressure of oxygen, so low amount of oxygen in the blood, so it's going to be sensitive to that. Not enough oxygen, not good. Sensitive to increased partial pressure of carbon dioxide, so more carbon dioxide also kind of means we're metabolizing too much, not getting enough oxygen, and the body doesn't like it. So decreased pH, so with increased carbon dioxide comes decreased pH, which is acidic. Okay, so increased carbon dioxide, it's the whole, CO2 plus water, carbonic acid, and then and that we'll talk about that a little bit more when we talk about pulmonary, when we talk about acid base. But increased carbon dioxide is going to ultimately lead to more hydrogen. Okay, so, and that's what it's detecting. Okay, so activation leads to activation of the sympathetic system. So everything that comes along with the sympathetic system, increased heart rate, increased contractility, and some vasoconstriction, all of that's going to take place. So we have increased pressure. All right. So central nervous system ischemic response. So ischemia is low blood flow. The brain is intolerant of that. It wants the blood flow to be what it needs to be, and if it's not high enough, it will shut down other systems and open up systems to get blood flow to the brain. Okay, so that's good. So chemoreceptors in the medulla, so again in the brainstem, the medulla, are most sensitive to high carbon dioxide and acidic blood. So low pH. Low pH is acidic. Someone many years ago decided that low pH means that it's acidic. And so really not measuring carbon dioxide or oxygen, but more in central detector, detection is more carbon dioxide, low pH. So results in sympathetic, again, activation to arterioles, controlling flow to those areas and redirecting it to the brain. So this is where the brain says, okay, forget everything else. Blood needs to come to me. Okay. All right, so overall increase in vasoconstriction, total peripheral resistance, and arterial pressure overall. So you have increased pressure overall, but you have redirecting of blood flow to the brain. Okay, so increasing how quickly blood is pumped improves longer-term oxygenation. So this is just kind of a note to keep in mind. I said it before. But if you have less blood, if you have less oxygenation of blood, if you kind of notice those things, then one way to get that done is to push it through the system more quickly, and that way it's going past the lungs more often, getting oxygenated more often, and that's kind of a way to get that done. So this happens if for low blood volume, if you have a patient with low hemoglobin or even low blood volume, then their heart's going to go faster. Their breathing maybe a little faster to kind of get that oxygenation taken care of and get that blood cycling through. All right, so let's talk about local control. So metabolism and local control, blood flow and blood supply. So one thing we know, and we can kind of piece this together, is that if you have enough nutrients, then there's really no reason for the body to start sending blood or send a lot of blood to that area. Everything needs a blood supply all the time, so we're talking about capillaries. So everything needs a blood supply, but how much blood is kind of a waste if you're sending a lot of blood to an area that has enough blood supply. So we can kind of look at nutrients, and the presence of nutrients will automatically cause constriction of those blood vessels. Okay? So by nutrients we mean oxygen. Okay? Oxygen is good to have. Glucose, so we have enough sugar going to those vessels, or to those cells. Amino acids, they're getting enough amino acids, and fatty acids. So they have these nutrients that they need, and so they're just like, okay, you can chill, you can take the rest of that blood somewhere else, we're doing good here. However, under stress, or when those cells are very, very active, let's say it's a muscle cell and it's very active, it's going to start building up waste products. So waste products meaning carbon dioxide, okay, lactic acid, because if you remember your Krebs cycle, all that stuff, you have glycolysis, and then if that turns into pyruvate, but if it doesn't, it turns into lactic acid. If you don't have enough oxygen, then it doesn't go into the mitochondria, and your Krebs cycle basically isn't activated. So you just have glycolysis and then the production of lactic acid, because that can happen without oxygen. Okay? So you have glycolysis can happen without oxygen, just happens out in the cell. The mitochondria is when you make ATP with oxygen. Okay? So buildup of lactic acid means that you don't have enough oxygen, and you're kind of doing this desperate anaerobic production of ATP. Okay? So that's what we're doing with that. I know that was a very brief description, but it's kind of an important concept, because anaerobic is making ATP quickly, but it's also has this byproduct of lactic acid. And it's also obviously an indicator to the body that there is something wrong, okay? Because it should be using the mitochondria and going through that whole Krebs cycle. All right, to make ATP, to make a lot more ATP more efficiently. That's really fast going through that. But anyway, so if we see lactic acid, we know that there's anaerobic respiration going on, and that's not the body's most efficient, best way to do it. Now, nitric oxide can also be produced from the breakdown of small amino acids. Adenosine, AMP, adenosine monophosphate, adenosine triphosphate, or I'm sorry, diphosphate, but not ATP. So if you're spending all your ATP, you may have a buildup of these molecules, adenosine, AMP, and ADP. And so that's also another indicator that you're using a lot of ATP. So that could say, all right, we need an increased blood flow. Decreased pH, so more acidic. Okay, also occurs with high metabolism. So all of these things together say, let's cause relaxation or dilation of the vessels, and that is going to increase flow. Okay. All right, so it kind of happens automatically. So, blood flow can increase 7 to 8 times as a result of vasodilation of arterioles and precapillary sphincters in response to increased rates of metabolism. So, when that metabolism is higher, we're going to dilate and get more blood there. So, vasodilator substances are produced as metabolism increases. So, that's kind of what we went through here. Nitric oxide is a major vasodilator. So, long-term local control capillaries become more dense. Oh, so long-term, when we're talking about this, when we're talking about this, the short term based on metabolites or waste products or nutrients, that's more of a short term solution from vasoconstriction, vasodilation. Long term, you can actually just put in more blood vessels. So we see that like with people who get in better shape. Their muscles may, if you use your muscles more often, then it'll cause something called angiogenesis, and you can grow new blood vessels, more blood supply, and you can kind of feed the need of those cells for of nutrients, and then more blood supply means you can get more oxygen and more other nutrients to it as well. Okay, so coronary blood flow. Want to talk about that. Coronary blood flow is the blood flow that's going to feed the heart muscle directly. So we have right off the aorta, so if we look at this picture and we see that this is the aorta here, right off the aorta there are little holes that allow the blood flow, before it even goes to the rest of the body, to move right into the vessels of the heart. Okay? All right. So that's going to provide nutrients to the heart muscle as it needs it. So it goes left and right coronary artery, and then those divide into the posterior descending, right marginal, and then on the left side, the left circumflex, left marginal, and then it continues to divide and feeds all the cells. So we don't have to get too much into it because really it's very similar to what we just went over. So blood flow through these vessels is almost entirely controlled by local metabolites. Very little autonomic control. So we're not vasoconstricting, vasodilating in the way that we saw with sympathetic or parasympathetic activation of these areas anyway. It's just that if there's more need, you have more waste products. If you have more waste products, you vasodilate. And you increase blood flow to those areas. That's what we mean by that local control, local metabolites. So hypoxia and adenosine are the primary factors. So low oxygen, that's what hypoxia is. Adenosine, we saw that with the adenosine monodiphosphate. And so those are going to indicate that there's a need. So increase in contractility. Well, so why do we care about this? So when the heart works harder, so when it beats harder, so increased contractility, that's going to lead to an increase in oxygen consumption, and that's going to cause the decrease in oxygen, which is going to cause vasodilation of these vessels. Now the interesting thing is that every time the heart beats, when it contracts hard like that, it kind of squeezes the coronary arteries and stops blood flow temporarily to these areas. So you have to have that compensatory that's kind of pumping through there because of the nature of the contraction of the heart, that it doesn't get this constant flow. It's actually kind of a turned on, turned off kind of a flow. But either way, we're trying to get enough oxygen, enough nutrients to deal with that as well. So local hypoxia leads to vasodilation of the coronary arteries, increased blood flow. Okay, so it kind of fixes that locally, just like we just saw. All right, so microcirculation and control blood flow. Now, in the body, everywhere in the body, whether it's the liver, the spleen, any of the skin cells, the lungs, any cells that are inside your body have to have a blood flow to them. Now, we know that we can increase, decrease. We saw how local control can do that based on the metabolites or waste products that are involved, the nutrients or waste products. And we see here that, and we've already talked about how pressure through the capillaries can cause fluid to move out and feed all these cells. So the cells are all sitting out here. That's our interstitial fluid. Okay, that's the fluid between the cells. Interstitial means between the cells. So when we're talking about blood flow and we're talking about blood pressure, one thing that we need to maintain is enough hydrostatic blood pressure. So we'll call that PC, pressure in the capillaries. So hydrostatic pressure is pressure in the capillaries. Okay? So that's capillary hydrostatic pressure. And when that's high enough, then overall fluid moves out of the capillaries and into the interstitium. Okay? And so it'll start to feed all of these cells here. So it carries, nutrients with it. And then, so the interstitial pressure at this point is very low, so it's not really pushing anything back. So this bullet here, it says there's hydrostatic pressure both inside and outside the capillaries because there's fluid in both places. If you have fluid in here, then that's going to push. And if you have fluid here, it's going to push back. But you can see here that there's hardly any fluid pushing back into the capillary. So your hydrostatic pressure pushing back into the capillary is pretty much, it's nominal. I mean, there's hardly any pressure pushing back. Now, but we also have proteins, okay? So we'll make proteins green. We have proteins in the capillaries, and sometimes we have proteins out here as well in the interstitial. But we have more proteins and blood cells and everything else, particles. We have more of those in the capillaries. Okay. So we have kind of an overall pull of fluid back against the hydrostatic pressure. So the hydrostatic pressure is pushing out here. We have numbers. So it's pushing out at 30, the hydrostatic. And the hydrostatic pressure from the interstitium is -1, so whatever. Okay, that's not enough to matter. So you've got overall 29 pushing out. 26 pulling in. So that means you have a net movement of fluid of a +3. Okay. Oh wait. No, that's a matching. So let's see. Did I do my math wrong? Right. So +6. Okay. So +6 is moving out because we have -26, but we have another +6, +3 here. Oh gosh, I'm not having a good time. So pushing out of the capillary is 30 +3, pushing out. So that means that there's a colloidal pull out of only +3. It's not much, but it's still pulling out. The hydrostatic pressure pushing in is -1 and -26. So all together, so you've got 33 -27, which means that you have a +6 overall pushing fluid out. So fluid is moving out. Okay? Those four things, it's important to remember those. Okay, so we have capillary hydrostatic pressure. Okay? We have interstitial pressure, hydrostatic pressure out here. And we can see that they're pushing and pulling. This is just pressure, fluid pressure, blood pressure. We have colloidal pressure, which is kind of a pull of, it's an osmotic pull. It's saying, wow, look at all these particles here. We have fewer particles out here. So we have this pull in of -26. Okay? And it's saying, and then there's always kind of a, even though this is higher, there's still some pull. There's some particles out here. And it's saying, okay, well, there's some particles. So it gives it a +3, saying, okay, we've got 3 trying to move out of the capillary. Okay? So those forces, that interplay between all of these pushing and pulling is what determines whether fluid is moving out. Okay? So you can kind of see with what we've been talking about, if our hydrostatic pressure is low, our blood pressure is low, arterial blood pressure is low, this number is going to be a lot lower. Okay, so, and we kind of see that over here. So we see that if our blood pressure drops, and now hydrostatic pressure from the capillaries is only 25, okay, and we still have this -1 pulling back in. But now we have especially if we have less blood, we've got our oncotic pressure pulling fluid back in from this interstitium out here of -32, and -29 overall. So we have ultimately more blood in this case, or more fluid in this case, is being pulled into the capillary. Okay, overall, that's what this means, is that if just basically by reducing fluid volume, and reducing blood pressure, we've caused a net movement of fluid from the interstitium into the capillary. So overall, big picture, the simple part about this is that when blood pressure is lower and fluid volume and blood volume is lower, we're going to start sucking it out of the interstitial from around the cells. We're going to start moving it from around the cells. That's not good for the cells, but it does keep your blood pressure higher. Okay? Or keeps the blood volume higher. All right, let's see, if more protein, so I have down here, if more protein is added to the blood, then that will increase capillary oncotic pressure. So that's going to maintain your blood pressure. So at least vital organs will have blood pressure, but it's going to be reduced but perfusion can still be reduced. All right, let's see. So reactions to hemorrhage, so we talked about that before with the baroreflex and increasing blood flow and blood pressure. We also see that renin-angiotensin increases blood pressure, and then we just looked at capillaries, how capillary flow can pull it from the interstitium, put it back into the blood, and keep that blood pressure high enough to at least feed the brain. All right, so that's kind of what's going on with hemorrhage. All of these things can be activated from that loss of blood pressure. So if the hemorrhage is, let's see, if your mean arterial pressure starts out at 100, that's pretty high, and then it's reduced, you have that blood loss, then you have all of these compensatory responses that we've been talking about that will get that blood pressure back up to a working pressure. So at least vital organs can get perfused. Or failure of the compensatory response, this is over time, 4 to 5 hours, then if it can't be maintained, then person's not going to survive. Okay, and then the last thing we're going to talk about, vasovagal syncope. So syncope is fainting, and vasovagal, vaso meaning vessels, vagal meaning the vagus nerve. So we have essentially what's going on is we have overactivation of the parasympathetic response. Somebody's heart rate drops, blood pressure drops because of blocking sympathetic activity. Their blood pressure drops and they drop, okay, basically. So reflex inhibition of sympathetic activity, in other words, a sympathetic event, so somebody may see blood, some people kind of freak out with that, they see blood and they pass out. Sympathetic event may cause magnified compensatory activation of the parasympathetic response. So even though seeing blood may be like, oh my gosh, what happens is the body will sort of reflexively overcompensate, and the sympathetic system will say, all right, stop, and will reduce the parasympathetic system will be turned up, reduce sympathetic activity, and cause a decrease in the arterial pressure and a decrease in, of course, cerebral blood flow, which is going to cause fainting. Okay? So activation of those medullary centers, increase in parasympathetic outflow, decreased heart rate, decreased cardiac output, decreased total peripheral resistance, decreased sympathetic activity, sudden fall in arterial pressure, and decreased blood flow. So sometimes, a vasovagal, so some people vagal their blood pressure drops when they're having a bowel movement. So like pushing. So a lot of times if a patient has like a high heart rate, you can have them kind of bear down like they're having a bowel movement, and that can kind of trigger this vasovagal response, and that can lower their heart rate. It can happen, but a lot of times people, when they have bowel movements, will faint. Okay, so that's kind of an outcome of that sometimes, because it triggers this vasovagal response, this vasovagal syncope. All right, and that's it. That's the last slide.